Hello, everyone. Welcome to today's webinar. I'm Kelly McSweeney, contributing editor at CNN Media, and I'll be moderating today's event. This webinar is titled Strategies for Advancing Molecular Glue Drug Discovery and is sponsored by WuXi Biology. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the top right corner of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will pose as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN online after the live webcast. Today's presentation is sponsored by WuXi Biology. As a global company with operations across Asia, Europe, and North America, WuXi Biology provides a broad portfolio of R&D and manufacturing services that enable the global pharmaceutical and life sciences industry to advance discoveries and deliver groundbreaking treatments to patients. Through its unique business models, WuXi Biology's integrated end-to-end services include chemistry drug CRDMO, biology discovery, preclinical testing, and clinical research services, helping customers improve the productivity of advancing healthcare products through cost-effective and efficient solutions. WuXi Biology received an AA ESG rating from MSCI for the fourth consecutive year in 2024, and its open access platform is enabling around 6,000 customers from over 30 countries to improve the health of those in need and to realize the vision that every drug can be made and every disease can be treated. During the presentation, we will hear from Dr. Peichuan Zhang, Dr. Lulu Zhang, and Dr. Jian Xiang. Dr. Peichuan Zhang was trained as a molecular geneticist and cell biologist with rich research experience using experimental models. He studied the mechanisms by which cells respond to stress conditions, such as ER stress and amino acid starvation, as well as their implication in diseases such as type 2 diabetes during his PhD studies. He conducted his postdoc work at UCSF, focusing on aging research using the nematode C. elegans, and continued these efforts to explore potential translational anti-aging routes in the alphabet company Calico. Since joining WuXi Biology, Peichuan has mainly been responsible for supervising teams and providing in vitro services to support drug discovery projects. Dr. Lulu Zhang holds a PhD in chemical biology and has conducted extensive research in cancer therapy. During her doctoral studies, she investigated the functions and mechanisms of breast cancer susceptibility genes through the regulation by small molecules. In her postdoctoral work, she explored the epigenetics related to hematologic diseases. Since joining WuXi Biology, Dr. Lulu Zhang has been responsible for multiple drug development projects covering various disease research areas, including oncology, and involving different modalities such as small molecules and peptides. Dr. Jian Xiang joined WuXi Biology in 2018 and currently works as the site head of the in vivo pharmacology unit at WuXi Biology. The team focuses on in vivo models and ex vivo analysis services, covering oncology, autoimmune, and anti-inflammatory diseases, among others. Beyond providing numerous different types of animal models, his team has developed many cutting-edge, unique models to support molecular glue drug discovery. I will now hand it over to Peichuan Zhang.
Let's get started. We would like to thank the organizer for the introduction. For today's webinar, we can talk about the strategies for advancing molecular glue drug discovery. The speakers include me, Lulu, and Jen, all from WuXi Biology. I will start by introducing the background, bringing out the concept and definition of molecular glue. Using this picture as an analogy, I try to highlight molecular glue is a kind of molecule that can stick two proteins together. And as a consequence, it can induce proximity to help stabilize existing interaction or create non-native new interaction, and also in some cases induce self-association. Based on the effects, molecular glues can be classified into several different types. In the case of inhibitory effects, there's type 1, for example, such as cyclosporine that can prevent or shield the native partner from interacting with each other. And then there's a second type that can redirect the interaction to a different part of the protein, such as taxol. And the type 3, such as lenalidomide, and that kind of immunomodulatory drugs, can produce novel activity. For example, polyubiquitination of protein by E3 ligase for UPS, proteasome degradation. About 30 years ago, it was discovered that cyclosporine, so this molecule here with a macrocyclic structure, a known immunosuppressant, can bring cyclophilin and calcineurin, this protein together into a complex. So this is perhaps the first report of a molecule that can work as molecular glue to stick proteins together. So this is a work from Stuart Schreiber's lab. And later on, people realized that perhaps there's even more compounds that could be intrinsically pro-type of molecular glues. By surveying the protein database, this publication actually shows that there's a lot of molecules known to induce heterodimeric complex interaction, either domain to domain or maybe motif to domain interaction, so classified into group 1 and group 2. So in other words, maybe we can find a lot of compounds that can be useful glues in certain applications. And with that said, we know that there's a lot of molecular glues such as cyclosporine, rapamycin, immunomodulatory drugs have been used in clinical trials have been used to treat human patients. There's even more actually have been tested more recently in clinical trials. So these glues target previous unligandoable proteins. I'm just showing a handful of them right here. So most of these are molecular glue degraders, MGD, so target different proteins. And most of them are also for oncology indication. But I also should point out that there's also non-degrading molecular glue, such as this one from Revolution Medicine for pan-RAS. And also there's a non-degrading covalent activator that target nerve2 keypole signaling. And also, Monte Rosa has been developing this molecular glue targeting VAV1, which is believed to be the first rationally designed molecular glue that can be used for non-oncology indication. So when we look back at the history of molecular glue discovery, at the very beginning, early stages, people probably just try to look for these compounds, molecular glue, pretty much by serendipity. So based on interaction between natural product and also native protein. But along the line, about 30 years later, people now can use certain strategy to rationally look for these compounds, to design them and test them. So I try to highlight, for example, we can start by using screening strategy to look for compound that can bind to certain protein of interest. And also, we can use different in vitro assays to validate this interaction, and also use functional tests to to ask for what will be the consequence, the readout, and eventually we can characterize the best hits in certain in vivo animal models. So these will be the subjects that will be discussed today in this webinar. And I'm using this slide. Basically, this is the workflow has been used by WuXi Biology to support specifically microglue degrader at a research stage. So using this box actually highlights at the screening part. So basically, we can use either affinity selection strategies such as DNA encoded library screening. Maybe we can also use other phenotypic assays that using conventional hetero screening strategy. So either binding assay, maybe look at the protein degradation efficacy assay, maybe other functional phenotypic readout. So to look for small molecules that can be the prototype to form as a prototype for microglue. Once we have found certain molecules that can bind to the protein of interest, so we would like to ask a question, which E3 ligase is engaged? And we want to do this so-called deconvolution step. And followed by that, this will enter into the routine structure-activity relationship campaign. So by doing different kind of orthogonal assays, biochemical, biophysical binding assays, we can ask, does the microglue bind to the, for example, E3 ligase? There's binary binding. Or does it bind to the E3 ligase as well as the protein target, so form this so-called ternary complex. And also, we want to perform cell-based assays to look at the degradation of the protein of interest. And we also, again, at several levels to confirm the formation of ternary complex. And also there's target engagements, selectivity, so on and so forth. And for certain degraders, we would like to perform structural biology to evaluate, for example, ternary complex formation. And some of them will be subject to other mechanistic studies. And also, eventually, certain compounds will be selected and further tested by different in vivo assays in different models. So this is the pipeline or the discovery workflow that has been used by WuXi Biology to help advance molecular glue drug discovery. And I want to touch actually briefly on this subject by how we're going to use high-throughput screening support at an early stage to assist molecular glue drug discovery. So here is one example using this affinity selection, again, so-called DNA encoded library DEL screening strategy. So the workflow is basically, imagine that we have a protein of interest. We can mobilize this target 1 to the matrix. And then there's a second target. Imagine this could be a E3 ligase, cereblon. So this second target will be pre-incubated with molecules, small molecules, tagged with different DNA tags. And when we mix them together, upon incubation, certain molecules can stick target 1 and target 2 together. So this is what kind of molecule we would like to look for by using this stealth screening strategy. So in the case of binary complex, so imagine that we can identify certain compounds. So there's an enrichment score. But when we have both the target 1, that's protein to be degraded, or we target 2, that's E3 ligase. So there's actually even more enrichment. So this is what we would like to look for. So such kinds of molecules can substantially increase the terminal complex formation. So at WuXi Biology, we have a diversity library that consists of about 15 billion compounds. We also have a cerebellum-focused library that consists of more than 5 million compounds. So these compounds, they have a structure similar to one that binds to cerebellum. So in other words, we have a large collection of library compounds that provide a very substantially expanded chemical space that are suitable for novel E3 ligase discovery, or maybe the search for entirely novel chemistry. So further downstream from the screening, we can imagine we should use a lot of different in vitro assays as the early stage for hit triage. So I may use VAV1 degrader glue as a showcase. So basically showing how we can prepare the reagent and engineer cell lines of interest to look at the degradation inside cell. And also using proteins, we can develop different orthogonal assays. And also there's consequently functional assays as well as target selectivity. And we also have certain in vivo models related to VAV1 and also other glue testing. So about the science, background of microglue target VAV1. So VAV1 encodes a guanine nucleotide exchange factor. So this gap factor is believed to promote GDP dissociation from the RAC GTPase, shown here in this simplified diagram. So VAV1 acts downstream of a T cell receptor as well as a B cell receptor signaling. And consequently, it functions as a key regulator of the immune cell development and activation. And it also has been shown to have a pretty strong impact on autoimmune and other inflammatory conditions. So at this stage, Monte Rosa has been actively developing a VAV1 microglue degrader. Shown in this simplified cartoon right here. So this degrader likely engage a part of the C-terminal and the RT loop of the VAV1 protein, and this is the cerebellum. So basically, this is a ternary complex revealed by cryo-EM. And also, this glue has been tested in phase 1 clinical trial that involved more than 70 healthy participants. So according to the results released from Monte Rosa, it has achieved more than 90% of degradation of the target protein VAV1 with strong suppression of inflammatory cytokines. And this is the microglue activity. So basically, we can appreciate there's a different kind of in vitro assays have been used to profile this specific VAV1 degrader. And in this webinar, we would like to use this as a benchmark to demonstrate that we can similarly develop a strategy or maybe the approach that can help characterize a microglue degrader. And this approach can also be applied to other microglue or maybe even like PROTAC degraders. So again, this is just the outline of early stage evaluation of the VAV1 degrader. We can generate the reference compound and the protein and use that to build up different assays. and evaluate degradation inside cell and also selectivity, so on and so forth. And the protein, we used the flag tag, the full-length Web1. So we use this protein, bacterially expressed, to build up some of the assays, binding assays, as described in the next couple of slides. We also generate the tool compound using a known reported Web1 degrader. So this part is basically the first question is, does the molecular glue degrader, does it bind to the E3 ligase of interest? In this case, is cereblon. So we are doing different orthogonal assays. Could be biochemical assays, biophysical assays, maybe cell-based assays as well. In the case of biochemical assays, shown here is we have a tagged version of cereblon. So the antibody, so basically is conjugated with the donor. So there's a receptor right here. It's a thalidomide red. So once there's an energy transfer here, there's a fluorescence produced. So one can imagine that if the compound can disrupt this interaction, maybe also disrupt this energy transfer, we can observe inhibitory effects on FRET signal. That's exactly what we have observed for this reported Web1 degrader. So it showed that IC50 inhibitory concentration, IC50 about 1 micromolar in this binary assay. Please keep this in mind. And likewise, we can use other assays such as biophysical SPR assay or MST assay to look at the binding. In this case, actually, it's a MC1. It's a BRD4 degrader against VHL complex. So furthermore, we can address the same question basically using cell-based assay. In this case, it's a nanobread, which is a type of assay that can analyze, help analyze target engagement inside live cells. So the principle is shown in this diagram. So basically, we can tag E3 ligase with nanolook, which can produce luminescence. And then this is a tracer can bind E3 ligase, which is typically a small molecule derivative. And then there's when there's energy transfer, so basically bioluminescence energy transfer, BRET happening, this molecule can produce fluorescence. So imagine that we treat the cells with certain compound, and the compounds can cause dissociation of the E3 tracer. In this case, we will observe inhibition of nanobread signal. That's what we have observed in this showcase. So it's a cereblon target engagement assay. So basically, in cells that have been transfected to produce nanobread E3 ligase, cereblon nanolook. When we treat the cells with lenalidomide or ARV, the Avinas AR degrader, which also targets cereblon, we actually can observe inhibitory effects to different degree, indicating there's actually target engagement by these small molecules, small molecular glue degraders. And the next question we want to ask is, if we have a molecular glue degrader that's also induce ternary complex formation, in this case, we can also use different kind of binding assays to address the same question. And in the case of Web1 and cereblon, again, we use the biochemical assay, which is also TerraFret. So shown here, we basically so the glue can stick these two proteins together, and when there's energy transferring happening right here, we would observe an increase of the TerraFret signal. That's what we have observed here. So in this case, this reported reference Web1 degrader can substantially increase the signal, HTRF signal, at a relatively low concentration. So remember, we actually have about 1 micromolar in the binary assay And now we actually have a much higher efficacy in this ternary complex, suggesting there's a cooperativity happening. And we can also use biophysical assays, basically to address ternary complex formation for PROTAC or molecular glue degraders. So this is a showcase for other targets. But similarly, we can develop such kind of a assay for the VAV1 degrader or the degrader of interest. And we can also use orthogonal cell-based assays. In one case, we can do this nanobit. So it's basically a split luciferase. So we have two proteins, one tagged with a large bit, which is a large part of the split luciferase. The other one is a small bit, which is 11 amino acid, which is N-terminal sequence of the luciferase. So when we have a certain molecule, such as molecular glue or PROTAC, and bring these two proteins into proximity, this large bit and small bit can complement to each other and form a functional luciferase and produce luminescence. So that's demonstrated here. So again, a Venus compound can help form ternary complex in this nanobit assay. Likewise, we can also use nanobread. So here is basically when we use the protein. So again, there's a nanolog tagged protein. There's actually another halo tagged protein B. So demonstrated here is basically we can use either wild type or the C terminals SH3 domain of the VAV1. So basically using these two, we can actually see pretty strong nanobread signal transfer. And also once when we use this mutant, so as reported in the science paper from Monterosa, so when we mutate the residues, basically that can essentially abolish such effects. So basically that is a two compound dependent breadth signal inside live cell. And so far we have talked about the different kind of binding assays, binary assay, binding, and the ternary binding. And then the next question that we like to address is, do the compound actually produce certain effects inside cell? In this case, it's efficacy degradation. So we can use either a transgene approach. So basically we can tag the endogenous VAV1 with a tag. So consequently, we can actually use a luminescence-based assay to detect degradation of the protein. I should say, this is actually a surrogate assay. We didn't really look at the degradation of protein per se. We are looking at the reduction in the luminescence and use that as a surrogate protein degradation. So demonstrated here, in Jurkat cells, when we look at the tagged VAV1, basically by incubating the cells for different time points, 4 hours, 6 hours, or 24 hours, we can actually observe this reduction in the luminescence, indicating the degradation of the protein. And again, using ATP level as a surrogate of cell viability, we demonstrated that, as reported, the tool compound actually didn't produce much effect on cell viability. So we can also use another approach. In this case, it's basically by using a Western blot. We can look at it directly at the level of the endogenous protein, VAV1. So again, in Jurkat cells, we demonstrate that upon 4 hours of treatment with the VAV1 degrader, it produces substantial degradation of the protein, the VAV1, as reviewed by the Western blot analysis. So it has a DC50 about 22 nanomolar against VAV1. And when we also look at the other gap factors, VAV2 and VAV3 of interest, so these are factors related to VAV1. So maybe in the case of VAV2, we observed a modest effects, reduction in the protein level when we use two compound at a relatively high level. And but overall, we didn't observe very strong obvious effects for these two compounds on reducing VAV2 or VAV3, indicating its selectivity against VAV1. So finally, I just want to say, because these are all in vitro assays, eventually want to come back to the biology. We want to do some functional assays. So particularly using primary immune cells. So here we are looking at activated T cells and B cells, and also using interleukin IL-2 level and the IL-6 level as the readout. So demonstrated here is that the reference compound can act to suppress IL-2 production and IL-6 production in activated T cells and B cells respectively. To this end, I have described using VAV1 molecular degrader as a showcase to basically tell you how we actually use basically different we develop and validate different kind of in vitro assays and use them basically as the approach to help evaluate molecular glue degrader. And then next, my colleague Lulu is going to talk about the non-degrading molecular glue. So Lulu will talk basically about the RAS and the RAF molecular glues.
Thanks, Peitran. For non-degrading molecular glue, there are actually different research fields involved, and today we will mainly focus on the RAS-MAPK pathway. In this pathway, the upstream receptor, such as tyrosine kinase EGFR, they may receive signals and then transmit these signals downstream. This step is regulated by GEF. GEF is short for guanine nucleotide exchange factor, such as SOS1. GEF activates RAS by converting it from an inactive GDP form to an active GTP form, and RAS is regulated from off-state to on-state. The on-state RAS or active RAS can further activate downstream effectors such as RAF or MAPK, promoting cell proliferation and survival. Active RAS is rapidly inhibited due to its intrinsic GTPase activity, and it goes back to off-state by this process. However, if RAS is mutated, this inhibitory process will be blocked, so RAS will remain in the on-state, continuously activating downstream pathways, and thus contributing to various diseases, especially cancers. RAS mutations are most single-base missense mutations, mainly occurring at codon 12, 13, and 61. These mutations are present in many cancers, with different frequency and subtypes. For instance, as we show here, in pancreatic cancer, KRAS mutations account for about 70% of all cases, with G12D being the dominant substitution. So KRAS G12D is a promising therapeutic target for pancreatic cancer, and such regulation is also suitable for other diseases. Different non-degrading molecular glues are involved in this pathway, and the first we are going to introduce is the RAF-MAC glue. For these two targets, there are actually lots of inhibitors discovered. Current RAF and MAC inhibitors, like trametinib, often come with side effects and resistance, making them less effective over time. For example, some RAF inhibitors work well in BRAF mutant cancers and can be bypassed by CRAF. Some inhibitors target BRAF CRAF heterodimers but can be bypassed by ARAF. So this highlights the need of a pan-RAF inhibitor. The NST628 is a non-degrading molecular glue that meets these requirements. NST628 combine to MEK and RAF through the alpha G helix form a ternary complex, and this complex will suppress the MEK phosphorylation and further inhibit the activation of the downstream targets within this pathway. So by this mechanism, this molecular glue can effectively overcome the bypass issue and shows great results across different tumor models. In addition, with better dosing and fewer side effects, the NST628 is effective on its own and an excellent partner for combination therapies. Moreover, another advantage of this glue is the permeability. So it's a potential treatment strategy for multiple diseases. And this non-degrading molecular glue is a promising candidate for regulating the RAS-MAPK pathway and related oncology treatments. Another non-degrading molecular glue is about RAS itself. Various inhibitors have been developed so far to target different states or mutants of RAS. One approach is a non-degrading molecular glue, and this glue can target active RAS. Cyclophilin A, also known as CYPA, is a cytosolic protein with high abundance, and it belongs to a family of isoforms. This protein was selected because its electrostatic surface charge matches well with RAS. A molecular glue can bind both RAS and CYPA, forming a ternary complex. This ternary complex will block the effector binding interface of RAS and then inhibit RAF binding to suppress the downstream pathway. And based on this mechanism, we have established the RAS-RAF disruption assay. This assay detects the FRET signal between RAS and RAF. When molecular glue is involved, the ternary complex, including RAS molecular glue cyclophilin A, can be formed, and this ternary complex will reduce the FRET signal. And while in the absence of cyclophilin A, no such inhibition is observed. So this indicates the RAS-RAF FRET is not influenced by the compound alone. This strategy is effective for RAS research. The RMC 6236 performs quite well in clinical research and seems a promising strategy for RAS-related cancer therapy. In conclusion, the non-degrading molecular glue is an effective approach to target regulation, and we are expecting more progress in different research areas. And this is the in vitro section for molecular glue, and Dr. Xiang will then introduce the in vivo part. Thanks.
Hi, everyone. This is Jian Sheng from WuXi Biology. Petra and Lulu have talked about the in vitro screening and evaluation of some molecular glue drugs. And for the rest of the talk, I will mainly focus on the latest stage discovery and translational research of this type of drug. Currently, researchers have attempted to take advantage of protein-protein interaction strategy in nearly all of the disease areas. Among them, oncology, autoimmune and inflammation disease, and CNS disease attracts the most attention. So far, many newly developed drugs are already in the process of clinical trials. Drug evaluation, especially the in vivo efficacy and the PK/PD study, is one of the key steps during the lead selection and optimization. WuXi Biology has accumulated a lot of experiences in multiple in vivo models. We have established more than 400 of CDX models, 1,500 PDX models, which both covers 30 cancer types. Most of the models are well annotated by SOC and profiling. In addition to subcutaneous model, we also established 6+ orthotopic models and 80+ metastasis models. And beyond the basic tumor models, drug resistance platform is one of our advantages. Utilizing different models, we have established more than 100 engineered resistant models, 30+ induced resistant models, and a series of clinical relevant PDX models. As to the autoimmune and inflammation disease, we have also developed more than 100 different types of models, which covers different indications like rheumatic disease, skin disease, systemic disease, intestinal disease, acute inflammation, and neuroinflammation. And our disease models contains not only rodent species, but also NHP and other large animals. And for the CNS disease, we have extensive experience in neurodegenerative disease, pain, neuropsychiatric, and other disease models. The mission of our in vivo model team is to refine models to reflect disease reality and test models to mirror human responses to different drugs. Then I would like to show some cases about the molecular glue evaluation. On the left column, there are two cases of oncology fields. The upper one shows that lemonidonate produced good efficacy in tested lymphoma PDX model. The lower one shows that MRT-2359 and molecular glue targeting GSPT1 also showed excellent efficacy in prostate cancer model at a relative low dosage. In the middle column, EAE model and T-cell transfer induced colitis models are two popular autoimmune disease models for T-cell related molecular glue drug evaluation. Figures show that these models provide a considerable therapeutic window for efficacy evaluation of the drugs. Except for efficacy, POI protein level change is an important PD result to demonstrate the MOA of the drugs. Targeted or non-targeted organs in disease models can also be collected for further analysis. Overcoming drug resistance is one of the key advantages of utilizing molecular glue drugs. Since drug resistance could happen to any type of drug and any type of target in patients, it is quite important and urgent to establish cell-based or animal-based resistance models to mimic the resistance properties and explore the potential solution to overcome them. Here, I would like to show our strategy to establish drug-resistant model. The first one is to obtain drug-resistant tissue from patient and develop corresponding PDX models. It is the most direct method, but resistant biopsies are not easy to get, especially for those ones from newly developed drugs. In vivo inducing in tumor-bearing animals is an excellent way to mimic the drug-resistant progress, and sometimes we're able to get several different models under different resistant mechanism, just like what happened in the patients. And it usually takes relatively longer time and great effort to obtain stable models. In vitro inducing in cells is another way with less cost. The inducing period depends on different targets and the drugs, but it is easier to explore the resistant mechanism with the cells. Introducing reported mutations or overexpression some target-related proteins into parental cells is another way to get a model with certain resistant mechanism. Here is a showcase of the KRAS-related resistant model. In order to obtain a KRAS G12C inhibitor resistant model, we kept up a AMG 510 sensitive tumor model and dosed a batch of tumor-bearing mice and continuously with the drug. After a long time, the dosing period, tumor tissue become drug resistant to AMG 510. You may see the drug sensitivity validation result here. Once getting the drug resistant model, we can utilize this model to test a new generation of KRAS G12C targeting drugs or other new therapies. Besides, we are curious about the drug resistant mechanism behind this model. Therefore, we carried out some PD validation and sequencing on different tumor tissues, and we are excited to find that the downstream of KRAS pathway and the tumor microenvironment are changed in resistant model. Furthermore, we are attempted to overcome the resistant phenotype using different strategies in this model. The first one is combination therapy of KRAS inhibitors and SHP2 inhibitors. We validate different clones from the tumor tissue with this strategy and found excellent efficacy results achieved in both clones. The second strategy is using a new pan-KRAS glue, IMC6236, to overcome the resistant phenotype. In vitro cell viability tests were conducted to compare the sensitivity of MG510 and RMC6236 in both parental and resistant cell lines. Interestingly, results show RMC6236 produce excellent viability inhibition in resistant line, which revealing molecular glue drugs can be an effective drug design strategy to overcome acquired resistance and was trying further in other targets and indications. Last but not least, Peitran, Lulu, and I are both from WuXi Biology. The whole WuXi Biology is providing a full spectrum of service and solutions in drug development lifecycle, supporting standalone and integrated projects from early discovery to clinical development. As a comprehensive discovery and translational biology center, we have nearly 3,000 experienced scientists all over the world and have extensive experiences in different drug modality, from small molecular, peptides, oligonucleotides, bifunctional moleculars, vaccine, and so on. And for the disease areas, we can cover oncology, immunology, infectious disease, inflammation, neuroscience, metabolic disease, and so on. Alongside with individual project request, WuXi Biology owns a specialized team to coordinate internally and support integrated service from hit finding to lead optimization and lead candidate selection with a highly efficient timeline and strong expertise in each individual area. If you are interested in WuXi Biology's drug discovery and translational service, especially the molecular glue topic we talked about today, do not hesitate to let us know. The contact information of our BD colleagues are listed on this page, and it covers different territories all over the world. You may also review our website and follow us on the LinkedIn or WeChat via scanning the QR codes. Thank you very much for attending and listening today.
Thank you very much, Dr. Peishuan Zhang, Dr. Lulu Zhang, and Dr. Jian Xiang for that excellent presentation. And we do have some questions from the attendees. And as a reminder to the attendees, you can continue asking questions through the Q&A box on your screen. The first question is, what are the advantages of molecular glues compared to PROTAC?
Yeah, this is Peitran speaking. Just to confirm, actually, I just want to say, so maybe I think one thing I want to say is compare with the PROTAC, molecular glues actually offer like a better, smaller size, and also better oral bioavailability, and also better cell permeability. So, and definitely there's one thing is like, because it's basically, it's a monovalent binding, and basically it can actually induce a lot of this new conformation that can be suited and explored later on. So these are something actually I have in mind, basically the advantage over PROTAC. Okay, thank you.
Thank you very much. The next question is, why do most researchers choose CRBN over other E3 ligases?
Hi, this is Lulu. Yeah, perhaps Lulu from Peitran Group, so I can answer this question from my understanding. Cereblon is really well studied on the E3 ligases compared to others. So it has multiple biochemical and structural data available, so we can refer to this information for drug discovery. And also, Cereblon has been successfully used in approved drugs, so it's reliable for protein degradation, already been validated in the whole process of drug discovery. So we can trust this Cereblon, this E3 ligase. And there are lots of Cereblon ligands out there, like lenalidomide, thalidomide. So this will make developing new degraders much easier, especially when we compare to other E3 ligases. And structure-wise, it's a Cereblon contains some small molecule binding site, and this will provide more structure or more information to the molecular design. So this is very helpful information for the researchers, I think. So this is why we can use cereblon over other Israel ligases. And of course, there are more and more Israel ligases have been attracting more researchers. So perhaps in the future, we can have more Israel ligases in the new drug discovery in this field. Thanks.
The next question is, how can the specificity be improved given the widespread use of CRBN?
So the question is, so why people choose? Sorry, actually, Lulu, can you address this question, please?
Yeah, sure. I think, just follow the previous question. Cereblon is widely used for the drug discovery in the field of molecular glue. And so how we improve the specificity is very important because we need to reduce the off-target effect. So from our understanding, we used to discuss this question, and perhaps there are several approaches to try. We need to in this research field, the G-loop of new substrate is very important. The G-loop is a 8 amino acid structure motif, and it contains some like glycine, which will be the binding site of cereblon molecular glue complex. And if we make some modifications of this G-loop, for example, we can design the molecular glue to recognize the side chain of or some other different binding sites of the G-loop. Perhaps this is one possible approach to improve the specificity. This is one approach, and then we can also engineer the molecular glue to engage the additional binding sites of the some other new substrates in addition to G-loop. So G-loop is very important, but some other binding sites are also available. So in this way, we can improve the specificity of the cereblon and other POIs. And in addition, perhaps we can also try to target some different cereblon residues. There are actually some key residues to form the hydrogen bonds with the G-loop backbone. And in this way, we need to set aside these key residues to pay attention to other residues. And in this way, perhaps we can have more diverse structure for the molecular glues. And perhaps we can have more different approaches to improve the specificity. And now these ones can be referred to. Thanks.
The next question is, what other E3 ligases has WuXi worked on besides CRBN and VHL?
Thanks for asking this question. So basically, we have been, the short answer is yes. So we have been working also with other E3 ligase. And right now, because I would say majority of the work so far has been done, including clinical work, actually has been focusing on cereblon. Because people somehow believe that maybe cereblon and decap and maybe some E3 ligase complex, they have been evolved because they're kind of doing this housekeeping, trying, basically this role to clear damaged proteins. So with that said, we also have tried to look for other E3 ligases. So for example, we have tried to use Dell screen again as a start point to look for basically ligandability for other E3 ligases. So far, we have actually, for example, we have found GIT4. So this is actually consistent with what has been reported. So that can be that E3 ligase actually shows pretty good ligandability. But if you want to know more details about this, maybe please just send us an email, actually on the screen, to WuXi Concierge. Thank you.
What is the drug-resistant mechanism of the model related to CRAS inhibitor? KRAS inhibitor?
Yeah, it's Jen speaking, and it's a very good question. There are several potential mechanisms behind this model. The first one is the amplification of KRAS gene. We observed the KRAS copy number again in the resistant model, and KRAS protein increased significantly after the AMG pharmacology dosing. The downstream of KRAS pathway, like the PERK, does not change much, even when we're treating the AMG pharmacology at a very high dosage. The second one is the tumor microenvironment of the resistant model is changed. Many chemokines and cytokines are upregulated in the microenvironment, and more myeloid cells are infiltrated into the tumor tissue, which will also contribute to the drug-resistant phenotype as well.
The next question is how to confirm the protein-protein interactions in vivo, but not only in vitro assays.
I think I can maybe take this question. It is relatively harder to have the result based on the in vivo protein-protein interaction, but we do have some assays, and we can collect the tumor tissue from the animals and do some binding assays, occupant assays, or just evaluate the drug and the tumor tissue at the same time to evaluate the protein-protein interaction there.
Next question is there any in vivo data that demonstrate the RMC-6236 can overcome the resistance in that model?
Yeah, it's we are validating some strategies to overcome the drug resistance phenotype in this model, and I didn't include all the data set here. One of our preliminary data has demonstrated that the RMC-6236 produced excellent in vivo anti-tumor efficacy in this model.
All right, that's all the time that we have for today. Any questions we didn't get to answer during the live webinar will be answered over email after today's live event. So thank you again to Dr. Peishuan Zhang, Dr. Lulu Zhang, and Dr. Jian Xiang for your fascinating presentation. And thank you participants for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you On24 for technology and production services, and thank you to WuXi AppTec for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Kelly McSweeney. Goodbye.